Hi guys, welcome to lecture 3 on the respiratory system. So far we've looked at the anatomy of the lungs and the oral cavity and the trachea, and now we're going to talk a little bit about um, gas gas laws and chemistry. And if you've had chemistry, you probably spent a little time working on the gas laws. They're interesting. They're pretty easy to do. Work with mathematically. And when we talk about the respiratory system, they're fundamental to understanding. So anyway, one of the first gas laws to be familiar with is Dalton's law. Dalton's law refers to the partial pressures of gases in a mixture. So our atmosphere is a mixture of gases. Um, the major constituent is nitrogen, about 78% nitrogen. Next in line is oxygen, about 21% oxygen. After that, the last 1% is made up of argon, carbon dioxide, carbon monoxide, and some other trace elements in that order, respectively. Um, and so when we look at a mixture of gases like the atmosphere, and we look at how gases go into solution in our blood, we talk about a concept of partial pressures. So in order to calculate a partial pressure, one of the things we have to take into consideration is what we call atmospheric pressure. And if you see here, there's a little diagram, and this is what we call a mercury barometer. And these things work pretty simply. You fill up a little tub with mercury, you stick a tube into that mercury with a cap on the end of it, and the air pressure of the atmosphere will press down on that and push mercury up into that tube. And it pushes it up to about 76 centimeters or 760 millimeters if you're at sea level and if the temperature is 25 degrees. So we have this concept in chemistry called standard temperatures and pressures. The standard temperature would be 25 Celsius, 72 degrees, and the standard pressure would be 1 atmosphere of pressure, or 760 millimeters of mercury, also called 760 torr. So there you go, that's what that means. And that pressure of the atmosphere, there's a column of gas above, just like if you were swimming at the bottom of a swimming pool, you've been subjected to a column of pressure, right? If you dive down to 11 feet, you can feel that pressure of the water column above you pressing down on you. Well, there's a column of gas above you, about 100,000 feet tall. Most of the gases in the atmosphere are concentrated in the first 30,000 feet. That's why airplanes have a tough time flying above that level. There's just not enough oxygen to burn. And that measured pressure, as I said, is 760 millimeters of mercury. Also is about 14 pounds per square inch, which is kind of interesting. It seems like a lot of pressure. Anyway, um, if I think about the partial pressures of that gaseous column, since nitrogen is 78%, then we say that nitrogen's contribution will be a total pressure of about 601 millimeters of mercury. Oxygen at about 159 millimeters of mercury. And CO2, which is our low concentration, less than half a percent, contributes about 0.3 millimeters of mercury. Well, that's important when we think about gas exchange, because remember that one of the central concepts in thermodynamics is this idea of diffusion. That is, matter likes to move from areas of high concentration to low concentration. And that's how we transport gases in the body. So in the alveolus, the O2 partial pressures are about 105 millimeters of mercury, and the CO2 levels are about 40 millimeters of mercury. And in the blood and the tissues, we have this differential. So we can see that oxygen wants to go into the tissues, and CO2 wants to come out of the tissues. And that explains our gas exchange from the perspective of Dalton's laws of partial pressures. So generally speaking, I've got relatively high oxygen concentrations in the alveolus and relatively low CO2 concentrations. In the blood, as it's brought to the alveolus from the systemic circulation. It's blue, not really, but it's illustrated as blue. It's got a very low O2 concentration and a very high CO2 concentration, so that allows for gas exchange here. As I pump blood from the lungs, now they've been richly oxygenated, out to those bodily tissues, I find tissues that are starved for oxygens, and I find tissues that have been creating CO2 through the process of cellular respiration, and that allows for the diffusion of CO2 back into the blood, and its removal subsequently by the alveolus. Now, if you recollect from our lecture on the pathophysiology of COVID-19, one of the things he talked about were these two types of alveolar cells. Type 1 are those cells that make up the alveoli, and they simply allow gases to diffuse through the membrane. Literally simple diffusion, right? O2, CO2 are small nonpolar molecules, and so they can just diffuse according to their concentration gradient through these cells. The alveolus is only one cell thick. The capillaries are only one cell thick, and so it's a relatively easy transition. Those type 2 cells are really interesting. They secrete something called surfactant, which is a fancy type of a detergent made in your body. And that surfactant helps break up surface tension in the alveoli and allows them to keep them open. So if you recollect from the lecture on that pathophysiology of COVID-19, once these type 2 cells are the target of COVID-19, so not only do you have this infection with this accumulation of edema, this fluid collection as a result of histamine release from the mast cells, but you also tend to destroy these type 2 cells, which decrease the amount of surfactant and increases surface tension in the system, and so that impedes gas exchange. In fact, if you're a newborn infant, one of the things that hasn't happened to you, if you think about this, if you're born before 9 months, your body's not ready to be born and particularly breathing oxygen. And so one of the things that's problematic for infants is the fact that those type 2 cells haven't become activated, and those infants aren't making enough surfactant to keep their respiratory system open, to keep the alveoli open. Tough being a kid. So we breathe in oxygen to do cellular respiration. And the way we carry oxygen in the blood is through a molecule called hemoglobin. So the red blood cells are called erythrocytes. And within the erythrocytes is a protein called hemoglobin. And if you recollect from our discussions on proteins, at the quaternary level of structure, most proteins are made up of multiple protein subunits, and that's the case for hemoglobin. It's made up of 1, 2, 3, 4 different protein peptide chains, alpha and beta chains. And then in the interior of these 4 subunits is a very cool little structure called the heme group. And the heme group, you'll see, has got a bunch of nitrogens incorporated into the cyclic or ring-shaped compounds. And at the very center of the heme is an atom of iron. So we say that these heme group coordinates iron, metallic iron ions. And iron likes to bind oxygen. It has a binding affinity for oxygen. That affinity allows oxygen to go from hemoglobin to oxyhemoglobin. And you can see here in this GIF that hemoglobin undergoes a conformational change when it binds oxygen. It also undergoes a color change. Why is your blood red? Well, FeO2, FeO3 likes to reflect red light. And so each red blood cell has about 300 million different hemoglobin molecules in there, and your red blood cell count is really important in your ability to perfuse your tissues with oxygen, that is, deliver oxygen to the tissues. As a side note, iron has a high binding affinity for O2, but it's not as high as its binding affinity for CO, carbon monoxide. And that's why carbon monoxide poisoning is so important. Once it binds, it outcompetes oxygen's ability to bind, and then you become anoxic and you die from anoxia. You don't have any oxygen. So, if you're burning things in your house, one of the products of combustion is carbon monoxide, particularly in an engine. And so it's very, very dangerous. Hopefully you've got a carbon monoxide detector in all your rooms, and they have good batteries in it. So hemoglobin binds oxygen. It carries that oxygen to different parts of the body. And when it gets to oxygen depleted tissues in the systemic circulation, it releases that oxygen. Of course, oxygen is going to obey, it's going to overcome its binding affinity for hemoglobin and follow its diffusion concentration gradient. And so there's a fine line between that binding affinity and oxygen's desire to come to equilibrium. Similarly, carbon dioxide is transported around the blood. And, if you've ever held your breath, you can do it right now. If you hold your breath, you'll start to feel uncomfortable. And you'll start to feel more and more uncomfortable. And I want to ask you to think about, what does it feel like? Are you dying to breathe in, or are you dying to breathe out? And the answer is the latter. And that's because you're making a lot of CO2. And CO2 does this funny little thing. A little bit of it's just dissolved in the blood. Some of it actually gets bound to hemoglobin in a structure called carbaminohemoglobin, but the overwhelming majority of it is turned into bicarbonate, the bicarbonate ion HCO3, and it's turned into carbonic acid, and that happens in the red blood cells. So every cell in your body is burning sugar and using oxygen to make CO2 and water and ATP, and that CO2 builds up. And the problem with carbonic acid is that it can change your blood pH. And so this bicarbonate ion system is a buffering system. It's a major homeostatic mechanism. And one of the things that allows you to store bicarbonate ion equitably and allow you to carry CO2 around is something called the chloride ion shift. When you hold your breath, you really want to breathe out. You want to get rid of that CO2. That's the thing that's working there. You have got a little pneumataxic center in your hypothalamus, and it's reading your blood pH levels. That's how it determines how well oxygenated, or more aptly, how much CO2 is there. And it wants to keep pH in this very narrowly defined value. Physiological pH is somewhere between 7.38 and 7.42. And so anytime you come out of that range, your body starts to do some radical things like make you breathe. And of course, you can change your blood pH by rapidly breathing or holding your breath. Anyway, there's this phenomenon called the chloride shift, and that helps maintain blood pH by compensating for the movement of the bicarbonate ions. So in gory detail, most of the CO2 is being carried in the red blood cells. And as CO2 enters the red blood cells, it combines with water to become carbonic acid through the action of an enzyme called carbonic anhydrase. Now, carbonic acid's unstable, and it immediately breaks down into the bicarbonate ion and hydrogen ions. And those hydrogen ions are actually bound by hemoglobin in the red blood cell. And when I go to deliver CO2, I need to kind of compensate for this loss of negative value, and that's when these chloride ions rush in. And so that's a chloride ion shift, and it's part of this bicarbonate buffering system. And I think that's all you need to know about the chloride ion shift. So remember that deoxyhemoglobin goes to oxyhemoglobin, and when that binds it releases hydrogen ions, and those hydrogen ions bind with bicarbonate ion, become carbonic acid. Which brings us to our review. So remember that most of the blood, oxygen in your blood is bound to hemoglobin, is oxyhemoglobin, and that iron is the element of the hemoglobin molecule that binds oxygen. These different partial pressures allow gases to move in and out, and then most of the CO2 is bound as bicarbonate ion.